let us begin by understanding the entire journey of a sample within a laboratory setting. We will explore the sample's complete life cycle, from its initial creation to crucial final decisions. Now, let us look at the detailed workflow for creating a sample in the laboratory. This process is vital for ensuring complete traceability from the very beginning. The first task is sample request generation, which initiates the laboratory process. We begin by initiating requests for samples from various sources and departments. These requests can originate from manufacturing, research and development, stability studies, or external customers. It is crucial to include essential details like product name, lot number, required tests, and priority. ALIMS, or Laboratory Information Management System, enables electronic submission, significantly reducing data entry errors. Next, we move to sample receipt and inspection, which is a critical verification step. Here, we carefully verify all the arrival details to ensure everything is correct. This involves inspecting the packaging and checking the seal integrity upon the sample's arrival. We must also check the transport temperature, often using data loggers for cold chain samples. Additionally, confirming that the quantity or volume matches the original request is very important. The next step is registration and test assignment, which organizes the sample within the system. We register each sample in the LIMS to ensure it is properly catalogued and tracked. The LIMS automatically generates a unique sample ID, ensuring no two samples are alike. We then populate mandatory metadata fields, either manually or through ERP integration. The system automatically assigns the necessary test based on the specific sample type. Following registration, we proceed to labeling and verification, a crucial step for identification. Here, we print the labels from the system and carefully apply them to the samples. The LIMS generates standardized labels, including the ID, a barcode, and other vital metadata. The analyst then scans the label to confirm it accurately, matches the digital record. Finally, we affix the label to a clean, dry container surface, strictly following standard operating procedures. The last step in this workflow is chain of custody initiation, ensuring a clear record. We meticulously log the initial custody of the sample as it enters the laboratory. The system logs the custodian's name, the exact timestamp, and the sample's storage location. This precise record forms the very first link in the sample's complete chain of custody. Let us now explore the essential sample metadata fields, which provide critical context. This contextual data is fundamental for maintaining both integrity and traceability throughout the sample's life cycle. The sample type is a crucial metadata field for proper classification. It determines all the required tests and how specifications are assigned to the sample. An incorrect sample type can lead to wrong limits or inappropriate tests being applied. The batch or lot number is another vital piece of metadata for tracking. This number links samples directly to production records, enabling recall traceability if needed. It is absolutely critical for investigating any issues that may arise during potential product recalls. The date and time of collection are also extremely important for accurate records. These details are required for accurate stability and hold time calculations in various studies. Well, incorrect dates can unfortunately invalidate the entire dataset of a stability study. The storage condition is a key metadata field, influencing sample preservation. This guides the proper storage at ambient, refrigerated, or frozen temperatures as required. It prevents sample degradation, thus ensuring that all analytical results remain valid and reliable. Now, let us compare different sample labeling technologies to understand their applications. Choosing the right technology is essential for ensuring robust traceability of your samples. First, let us consider the 1D barcode, a widely used labeling method. 1D barcodes offer fast, low-cost scanning, making them very efficient for many labs. However, they have a low data capacity, limiting the amount of information they can store. A significant limitation is that they can smear easily, potentially making them unreadable. They are best suited for routine samples where high data density is not a primary concern. Next, we have the 2D barcode, which offers enhanced capabilities. 2D barcodes are compact and can store significantly more data than 1D barcodes. A drawback is that they require a specialized 2D scanner for reading the information. These are ideal for high-volume laboratories that handle many samples and need more data. They also offer better durability, making them more resistant to damage or smearing. Then, we have the QR code, which is becoming increasingly popular. QR codes are highly convenient as they are scannable with most modern smartphones. However, their use may require a smartphone or camera, which might not always be available. They are particularly useful for field sampling, allowing quick data capture on site. QR codes provide versatile access to information, making them adaptable for various uses. Finally, let us look at RFID, Radio Frequency Identification Technology. RFID allows for scanning without needing a direct line of sight, which is a great advantage. The main limitation is the high infrastructure cost required for implementation. This technology is excellent for automated freezers, enabling efficient sample management. RFID tags enable real-time tracking of samples, providing constant updates on their location. Let us now examine the typical laboratory sample handling flow. Step by step. The process begins with receipt and verification, ensuring samples arrive correctly and safely. Next is sorting and prioritization, where samples are organized based on urgency and type. Following that, samples may enter a temporary holding area until further processing can begin. Then, we have storage assignment, where each sample is allocated its appropriate storage location. Preparation and aliquoting involves readying the sample for testing, often dividing it into smaller portions. Finally, samples are distributed to analysts, ready for their specific laboratory tests. Now, we will discuss the critical sample storage conditions that are essential for quality. Maintaining these conditions is vital for preserving sample integrity from receipt right through to disposal. Ambient storage refers to keeping samples at typical room temperature. This is typically between 15 and 25 degrees Celsius for items like tablets and solid raw materials. Refrigerated storage maintains a cooler environment for temperature-sensitive samples. This is usually maintained at 2 to 8 degrees Celsius for materials such as serum and reagents. Frozen storage is used for samples requiring colder temperatures for preservation. Samples are typically preserved at -20 degrees Celsius, commonly used for stability samples. Ultra-low storage provides an even colder environment for long-term preservation. These are stored at -80 degrees Celsius for long-term biobanking, DNA and RNA. Cryogenic storage represents the coldest conditions for highly sensitive biological materials. This is maintained at -150 degrees Celsius or in LN2 for cell lines and tissue. Let us explore the vital role of LIMS in sample handling and processing within the lab. LIMS helps streamline tracking and significantly prevents errors throughout the workflow. Barcode scanning is a key LIMS function that enhances sample management. It precisely tracks every single sample movement as it occurs within the laboratory. This feature ensures that the correct sample is always retrieved for its intended analysis. Inventory management is another crucial function provided by the LIMS. It displays a real-time count of all samples currently residing in each storage location. This system maintains a precise and up-to-date map of all sample storage locations. Chain of custody logging is automatically handled by the LIMS, ensuring accountability. It automatically logs all changes in custodian with their respective timestamps. This provides an unbroken and fully auditable record of every sample transfer. Automated alerts are a proactive feature that helps prevent potential issues. The system notifies staff when samples are approaching their designated expiry dates. It also flags potential hold time violations, which helps to prevent future problems. We will now discuss data entry and verification methods, which are crucial for accuracy. These methods are designed to ensure the highest level of accuracy in all laboratory data. Manual data entry involves analysts directly inputting results into the system. Analysts type the results into LIMS forms or various electronic worksheets. Automated data capture directly transfers results from instruments to the LIMS. Instruments integrate with LIMS to directly transfer all analytical results. The hybrid approach combines elements of both manual and automated methods. It combines automated data capture with mandatory analyst verification for added accuracy. Data validation rules are enforced by LIMS to maintain data quality. LIMS enforces range checks, mandatory fields, and specific format rules to prevent errors. Verification and review involve multiple layers of checking before data release. This involves a multi-level review by the analyst, a peer, and QA before final release. Let us now examine the key quality control decision types made in laboratories. These decisions formalize outcomes, ensuring consistent product quality and compliance. A release decision confirms that a product meets all required specifications. This decision confirms that the product meets all specifications, authorizing its next stage. A reject decision indicates that the product fails to meet the specifications. This indicates the product fails specifications, requiring reprocessing or even disposal. A hold decision means the product is temporarily paused for further assessment. The product is placed on hold, awaiting additional data or further clarification. A retest or investigation decision is triggered by unexpected results. This is triggered by OOS or OOT results, mandating a thorough root cause analysis. To summarize, let us review the most important takeaways from our discussion. Here are the key points to remember from today's comprehensive lecture. Robust sample creation and precise labeling are fundamental for data integrity and comprehensive traceability. Proper handling, secure storage, and LIMS tracking maintain sample viability and audit readiness. Accurate data entry and rigorous verification processes prevent critical errors in analytical results. Formalized quality control decision documentation or QCDD ensures scientific and regulatory compliance. Usage decisions are critical for product safety, consumer protection, and organizational credibility in the market. LIMS automation, continuous training, and best practices significantly drive laboratory efficiency and quality.